Hello, welcome to El Zoominario. Today we have Maria Garcia Mendoza. She was born in Lima, Peru, and emigrated to Iowa while still in high school. She has a bachelor's degree in biological research from Loras College in Dubuque, Iowa. She has a PhD in cellular and molecular biology from the University of Wisconsin-Madison, where she studied the interplay between immune cells and breast cancer. She also studied thymus development at the MD Anderson Cancer Center in Texas as a postdoctoral fellow. Currently, she is a scientist at Luminex Corporation in their Amnis Imaging Cytometry Applications team in Seattle, Washington. Thank you very much, Maria, for being here with us.
Thank you, Claudia. It's nice to share a little bit about what I'm doing. So I'll give you a little background about the science behind the project and also the analysis, which is the part that when I came in. And this is very different from academia, where in academia you do everything yourself. You collect the data, you do analysis, you publish it. Whereas in biotech, where I'm at now, you work with a team. So part of a team can collect the data, the other part can analyze it, another part can reanalyze it, which is what I'm doing here. So I'll show you that small part. Is it moving? No. There we go. Okay, so first the background about imaging flow cytometry. So this technique combines flow cytometry and imaging into one. And this is what the Amnis group at Luminex is what we do. So what is flow cytometry? So flow cytometry is a technique that we use in the lab, especially in immunology, to analyze cells that express things on the surface or also inside. So think of it as your blood. So all the cells have to be in a liquid state, in suspension is what we call it. So you have all your cells in suspension. They are single cells, so they're not big clumps. And you can label the cells with a fluorescent marker. So the fluorescent marker will be tagged to something like an antibody or a marker that would tell you what type of cell it is. So this big mix goes into the flow cell and goes towards an interrogation point. So once it gets to the interrogation point, the fluorescently labeled cells will have some light shining to it, so a laser from all different kinds of wavelengths, and then a light will be reflected. And you can collect that light, you can quantify it, and also draw conclusions as to what the cell expresses, how many cells express this marker, how many cells express the other one. So it's very useful quantitative technique called flow cytometry. So flow cytometry, as a recap, is fast, it's sensitive, quantitative, but you don't have a spatial resolution, which is like, where are things located? So if you look at my face, you know where my eye is, but when my eye and my nose and my mouth are all in liquid, then you don't know where things are. And that's why people use microscopy. So microscopy, everybody's heard of microscopes, I'm hoping. Very easy. You have a light source, you have a lens, so you can look through and magnify things. So very tiny things can be seen in full, such as over here. This is one of my images from my postdoctoral research, where we're looking at a structure. And here I wanted to quantify how many cells express the red marker, how many cells express the green marker. So this is very easy to collect, but it's kind of hard to quantify. So you need a really powerful analysis software, or you need counting with your eye. So it takes a lot of time to get information out of these kind of images, although they look beautiful. So microscopy, again, it's ultra high resolution. You can see very, very, very tiny things. And then the data is a little less quantitative, and it likes throughput. So you can get less data from a lot of time. And now we have the imaging flow cytometry, which is what my group does. And it combines both of them. So it's fast, it's very sensitive. You can quantitate all of the data, and it gives you a special resolution. So where are things at in the cell? This is a little small background of how the instrument work. It's pretty close. It's pretty the same thing as a flow cytometer. So you have your cells in suspension in the flow cell. You come to the interrogation point, a laser shines, and then you can collect all the information from it, all the light reflected. So it goes through a lens, like in a microscope, because you need to see it, and it gets bounced off between mirrors and filters so that you can separate all their information. And then it comes to this spectral decomposition element, which will separate all the images and all the light information from your different markers. You can see it here. So here's one cell that has 1, 2, 3, 4, 5 different markers that they're all in different spaces and different places in the cell. They all signify different things. So this is a marker that's inside the cell. This is a marker that's around on the cell membrane. And over here, this is a marker that has the nuclei, so the center of the cell. So it all depends on what cell you have, what cells express what. It's very interesting. So you can separate these signals, and our detector will integrate all the images that come together, and it's called a time delay integration. So this is happening super quick, super fast, but it gets slowed down at the detection point where you can merge all these images and you can get a nice crisp image. Because if it's going super fast, you think, well, it's never on focus or it doesn't look good, but our instruments can capture all the data fast. So there's a lot of applications. So we can look at cell signaling, internalization. So when something goes inside the cell, co-localization, so things matching up with each other, all cell cycles. So the one that we'll be looking at is cell-cell interactions, how two cells interact with each other. So imaging flow cytometry can enable applications that cannot be performed by either flow cytometry or microscopy alone. So now the analysis of the immune synapse. The immune synapse right now is very important. As we know, we're in a pandemic, so a lot of the things about viruses happen. So I'm going to use a virus example where we have the immune cells or immunity and activation. So the virus gets into the body and the cells, and this is called an antigen presenting cell, or an APC is what I'm going to call it. It gets exposed to the virus and it gets internalized. So the virus goes into the cells and it gets processed by the cell. So this is called antigen processing. So an antigen is a part of this virus that will create an immune reaction. So it gets processed and then it gets exposed to the cell surface. So it gets exposed right out here, and we can see this with our instruments. So when antigen presentation happens, this APC that's showing this viral antigen can be detected by a T cell. So T cells are the cells in the body that will activate and are more like professional immune cells. So they will identify this antigen and create a massive immune response. And the immunological synapse is the highly organized structure that forms when the antigen presenting cells are interacting with the T cell. So it's the space in between the two cells. And there's a lot of things that happen here, receptors, signals, and a lot of things that happen here in biochemistry that we can study. But first we need to identify that these two cells are making contact and that it's stable. And when that happens, then the immune response can happen, and we know that signals can be exchanged. So this is how it looks like. Here are cells. We have an antigen presenting cell here in green, and these are Raji cells incubating with a toxin, staphylococcal enterotoxin B, SEB. So a toxin just to stimulate the immune response. And then we have the T cells, which are in orange. And these were isolated from blood, from human blood. And once they are incubated for 45 minutes, you can see that they're forming this structure. And over here in the middle, the area of interface is what we call the immunological synapse, the immune synapse. So these cells, to see them, we label them. So CD3 is the marker that will label the T cells, and CD11 is the marker that will label the B cells, or the antigen presenting cells. We also use another dye called phyloidin that will recognize actin, and actin is a structural protein that's also important and localized here to this immune synapse, and DAPI, which is a nuclei. So we collected this in one of our instruments in FlowSight, and now let me walk you through the analysis. And the big question for this project would be in a lab, for example. So what percentage of cells that are exposed to something, this case was a toxin, it could be a virus. So how many of those cells can induce or form an immune synapse? This is the question, the overall question we want to answer. So for the analysis, we have our own software. So I'm going to walk you through it. It's kind of tedious, which is why now I'm using I'm using Amnis AI, so artificial intelligence, to analyze this data faster and better. So here we use features that are depending on the shape and size of the cell to differentiate them. So we have, for example, here we go. In this group over here, we have cells that are single cells. Remember, a conjugate, so two cells together, are two cells. So this wouldn't work. Then we have the next group, which is over here, which we can see that these cells are clumps. So that's also not a conjugate. And finally, our region of interest, where we see two cells that are together. This is most likely where the cells in immunological synapse are in. So the step 2, we need to look at the doublets. Remember, it's one T cell and one APC. So we look at the intensity of the signal to see which cells are expressing both. Again, here we see just one, or T cells. Over here, we see just one color, or green, which are APCs. And here are two colors. So here we can see that this is where most of our cells of interest are. The next step is looking at cells that only have one APC, so one cell in green, which is most likely this region, which has one cell in green, and you can see some of them have more than one T cell in orange. And you can see that our analysis is working well because you can see the images where down here we have two APCs. So then we need to now remember, now we have one APC in green, now we need one T cell in orange, and here's where they are. So again, this is looking at features, so the cell size, the cell color, how much intense is that color, and that can help us identify the cells populations. And here you can see that we're excluding those cells that have more than one cell. Another thing we use, as mentioned, actin is the structural protein, and we look at cells that have this very, very strong signal of actin in between the two cells. So we use DAPI, which is our cell nuclei, to make a mask of a region we want to quantify. So we make the mask is right between the two cells, so we know we're looking at the right spot. And then we look at the intensity of actin in that spot. And that way we can tell, well, this is most likely where the synapse is occurring. So we can see also that there are some cells that can have very, very close connections, but that doesn't mean that they're in synapse. That means they're very close, not there yet at the time of us asking the question. So we can look at colocalization. So colocalization are where two things are together in one place. So when you mix green and orange, you get yellow. So we're quantifying the yellow region. So here you can see there's very, very little yellow, barely no yellow, and here is yellow. So we use that too as another marker. So now you put the two together, the actin signal in the red and the yellow signal from the colocalization. And we have in this little corner cells that are not in synapse. And since we can see all the images, we can tell, well, this worked pretty good. And over here, we have all the cells that are in synapse. So again, we have really, really bright actin in red and yellow happening, which means that yes, most of these cells are in synapse. And the answer to our question, remember, what's the percentage of cells in synapse? So when you treat them with a toxin, we have about 16% of cells in synapse. Our control, which was treated with, it wasn't treated at all, regular T cells, only 3%. So indeed, this toxin does activate and form an immune synapse. So because we can see all the cells, this is where I come in, and I'm looking at all of the cells that we've collected, and they look pretty good. But if I keep looking for more, I still see that this is not very optimal. There's still some cells that have, again, two APCs or two T cells. There are some that you can see there's 3 cells here, but you don't. Some of them don't have the marker. So this analysis is not great. So what do we do now when we know that we don't have a perfect analysis? Well, now we use artificial intelligence. And our software group launched this last year. So I've been involved in the testing, and now we're coming with a second version. So I'm doing more testing. Just a little background, artificial intelligence is more of an umbrella term that describes the software that can learn from data to achieve a specific task. And it simulates human decision making. And what we use, it's called convolutional neural networks. And this is driven by powerful algorithms where you teach the machine to think. So I'm going to teach the machine what's an immune synapse, what's not a synapse, what are two cells, or what are more than two cells together. So we want to exclude that. And a neural network that we use is a deep neural network that is based in kernel processing and is well suited for image data classification. So this is details that this is the reason why we picked this one, because it's good for imaging data, which is what we have lots of. So we create an Amnis AI model specifically for the immune cells. So first, I combined lots of data to teach my software. So data from 5 different experiments. And I can select my truth population. So I use 3 in this case, no synapse. So it's the 2 cells like this, immune synapse, the 2 cells together, and more than 2 cells. So it's when we have a clump where you have 2 or more T cells, 2 or more APCs. So then I created my model and I taught the software what was what. And I used a training set and then applied this data to my control and my toxin treated samples. So these are the results, part of the result that it gives you. So this is a confusion matrix that mainly gives you the probability that the software will correctly identify your data. So for example, here it tells me that my model at this time has a 92% chance that it will give me a correct classification of immune synapse. And then we move down, so 93% that it will correctly identify cells that are not in synapse, and 83% of more than 2 cells. So it's not great yet. So I'm still working on it, of course. And then we have some measures of how good is this model? Should I use it? And we have information and precision. So it's measuring the false positives, recall, which is a measure of false negatives, and F1, which is measure of accuracy. How accurate is this model? So at this point, our model is 91% accurate. So of course we want to get to 100%. That's the best. And that we can get there when we have more data and we have better data that clearly teaches the instrument or teaches the software what is what. So it's a work in progress, but it's at least better. So lastly, how do I test the model? So I used experimental data, and this is a dose response curve. So we added toxin an increasing amount. So if you're adding an increase in amounts, you're supposed to, or you would hypothesize that you have increasing amounts of immune synapse forming. So the Raju cells were exposed to different amounts of SCB. Again, they were incubated with the T cells, and this is what we get. This is all classified with Amnis AI and showing the percentage of T cells in the immune synapse. So again, as this supports the hypothesis that with increasing amounts of the toxin, we do have increasing percentages of the immune synapse. So our model at least is tested and it works as it should. So with that, I'm just going to summarize. So imaging flow cytometry offers a quantitative analysis, and we can analyze large samples along with the information that a microscope can give us. Our flow cytometer flow site can measure the immunological synapse and the formation of T cell and conjugates, and this can be quantified using ideas with a long, very, very long analysis, or we can use Amnis AI that cuts in time a lot and also supports the accurate assessments of T cells and APC conjugates, which are indicative for an immunological synapse. So indeed, this software saves a lot of times. It can handle lots of data, so I like using it. And with that, I can like to thank my boss, Haley, and Sherry, who was the one that did the first analysis and ideas, and Brian is the person of the head of the software group that created the Amnis AI software. Okay, and with that, yes. So this is not as fun as I wish it would be, but The question that I want to ask and maybe start a little bit discussion of is the question, where are you from? And with time for me and my family that we're coming from a different country, it just becomes more and more difficult to answer this question because, well, we're from a lot of places. So when you ask me, where are you from? Well, I can say, well, I was born in Lima, Peru. And that is true. So people are like, oh, so have you been to this place in Lima? I'm like, no, no, no. I actually grew up in Pisco, Peru, which is 3 hours south of Lima. So I really don't know much about Lima, but I'm from Peru. So they're like, oh, okay. So do you go back and see your parents a lot? It's like, no, my parents live in the US. So my parents are in Clarion, Iowa, and that's where I went to high school. So it's like, oh, okay. So that's where you went to school. And it's like, yes, I also went to school in Dubuque, Iowa, which is 3 hours west of Clarion. So when you start talking about the Midwest, a lot of people ask me about Iowa and corn. And it's like, well, no, no, no. I actually spent a lot of time in Madison, Wisconsin. That's where I went to grad school. And that's where I met most of my longtime friends. And Madison to me is what I would call home because I've spent the most time there and have greatest memory of Madison. So now that I'm in Seattle, which is where I live now, I talk about the Midwest a lot, and somebody will chime in. It's like, well, aren't you from Texas? I'm like, well, yes, that's where I lived because I did my postdoc in Texas. So when everything gets combined, like I have so many answers for this question, where are you from? And sometimes it's tedious, sometimes you have to decide, and it's very important for the Latin culture because a lot of us were not born here. A lot of us have heritage from Latin America, so it's a hard question, and that's what I said. I would like to maybe have a discussion or foment some discussion of like, how do you want to answer this question? And what I came up with at the end is when they ask you, you can always ask, well, what do I want to say? What is my home? So like I said, home for me would be Madison. Or you can ask the other principal, how much do you have? How much time do you have? Because I have a long time to answer this question. And then some people ask you, well, you tell them, oh, I'm from Seattle. And I could say that. And they're like, oh, but you have an accent. So like, where are you really from? So that is kind of questions that come up. Or you can just say, well, no, I don't want to answer. What's the next question? Nowadays where you come to places where you network, and one of the icebreaker questions are, where are you from? And you just don't feel like answering, you can ask for another question. So that's my little bit about us as the culture now that have many places where we've been and many experiences, and makes us who we are. But it's very interesting because this question, there's not one answer. And yeah, it depends how much time do you have. Like, I think I just spent like 4 minutes explaining where I'm from, but it's very different for all of us. So thank you.
Thank you, Maria. I'm going to stop the recording now.